Hi, this presentation is about the structural basis for subtype-specific inhibition of the P2X7 receptor. The P2X7 receptor has been related to chronic pain. Chronic pain is a major public health epidemic, affecting more than 10% of adults worldwide. Afflictions include rheumatoid arthritis, persisting headaches, and lower back pain. While some people are treated with commonly used analgesics, many are resistant to treatment. The P2X7 receptor is an arising target for treating chronic pain, since chronic activation of the receptor brings about pain. It is predominantly expressed in the immune cells in the blood and the brain. Over the last 10 years, a lot of effort has been made towards developing a number of structurally specific P2X7 drug antagonists, which blocks P2X7 activation to alleviate chronic pain. However, mechanisms of action for these drugs are poorly understood, obstructing the development of effective medicine for human patients. In this presentation, I'm going to talk about the structure of the P2X7 receptor, its singular novel drug-binding pocket against 5 structurally unrelated antagonist compounds, and the specific conformational change during the receptor's activation. P2X7 receptors are trimeric ligand-gated ion channels which take part in extracellular signaling via ATP. Although P2X7 belongs to the P2X receptor family, it has many features that are different from the other P2X type receptors. For example, the P2X7 receptor requires a higher concentration of ATP than is required for other P2X subtypes for initial activation. Its channel activity is promoted by a longer duration of ATP applications, and it opens a membrane pore for molecules up to 900 Daltons to permeate. While analysis of crystal structures through X-ray crystallography displayed universal mechanisms such as ATP binding and gating throughout other P2X subtypes, some discrepancies remain. For example, structurally unrelated drugs seem to antagonize the P2X7 subtype only, but not the other P2X subtypes. Hence, it is important to look into the P2X7 receptor in detail. Using X-ray crystallography, the binding site of the antagonist receptor was mapped, and through electrophysiological experiments, it was proved that those inhibitors allosterically avoid activation conformational changes of the P2X7 receptor. There were 5 structurally unrelated antagonist compounds tested against the receptor, as shown in Figure B on the right. Unexpectedly, all 5 of them are found at the same drug-binding pocket between neighboring subunits, in proximity to the ATP binding pocket. This drug-binding pocket is surrounded by 13 residues, consisting mainly from beta strands in the upper body domains of the neighboring subunits. Drug binding is mediated by hydrophobic interactions between the drugs and the amino acid residues, especially at positions deep within the cavity, involving phenylalanine 95, phenylalanine 103, methionine 105, phenylalanine 293, and valine 312. Drug receptor binding interactions was further validated by site-directed mutagenesis on the drug-binding amino acid residue. Yopro-1, a fluorescent dye, was used as a proxy compound for receptor activity. It was found that residue F103 played the most crucial interaction with the antagonists. Binding of the tested antagonists to a site other than the ligand binding pocket infers that those compounds acted as allosteric non-competitive inhibitors. To elucidate the working mechanism for each drug, the dose responses were measured for the P2X7-mediated proxy Yopro uptake in the presence of each drug at multiple concentrations. An artificial but potent agonist BZ-ATP was used in these experiments to achieve saturating responses in the presence of the antagonists. For each drug, dose-response curves fit with the non-competitive inhibition model but poorly with the competitive model. Furthermore, shield plots showed nonlinear relationships, especially at higher concentrations, consistent with the non-competitive mechanism. These experiments suggest that all of the 5 tested drugs are allosteric non-competitive inhibitors. The intersubunit cavity formed by beta strands 13 and 14 in the upper body domain is much wider in P2X7 than the other P2X subtypes. To discover the involvement of the intersubunit cavity in P2X7 receptor activation, the movement of the cavity residues were monitored in the presence and absence of ATP. This was done by creating a series of cysteine mutants in the drug binding pocket, followed by measuring the channel activity mediated by ATP after introducing a cysteine reactive agent, MTS-TPAE. The cysteine residue was modified by adding bulky chemical groups to it. The bulky groups will supposedly hinder conformational changes during channel activation, resulting in lower channel activity. When MTS-TPAE was applied in the absence of ATP, 4 of the modified cysteine residues displayed irreversible current reduction. This is implicit of the notion that the covalently bound MTS-TPAE at those positions obstructs the conformational changes required for channel activation. Conversely, when MTS-TPAE was applied in the presence of ATP, the cysteine mutants did not show a significant current reduction. These results indicate that MTS-TPAE is more accessible in the drug binding pocket in the closed state, rather than in the open state. Putting it all together, we can conclude that the drug binding pocket narrows when ATP activates the ligand channel, validating the fact that such a conformational change is required for P2X7 channel opening. Detailed study and analysis of the P2X7 receptor drug binding sites done by the researchers of this presentation has the potential to develop clinical efficient drugs that would help alleviate the suffering of people worldwide from chronic pain. Thank you.